working in I don't know, theoretical biophysics and statistical physics. Is that okay? Is very famous and for all the work he has done in connected information theory and neural codes and neural computation, and more recently working in more at large in biology and the role of information in biology. So this has been extremely, all his works have been extremely influential in the modern modeling of biological systems. He's also been involved in education projects, so there are a lot of students connected online and here. So I think it's also nice to hear a person who has thought about how to convey information to the young generations. And today, so it's here for the day, and so in the afternoon, if somebody wants to have a brief chat, there is this possibility at the third floor of Rengen building. And so today, there were various possibilities for his talk, and at the end we converge on the idea of listening to how recent results, experimental results in neuroscience can help in modeling the neural system, generally speaking. But I will leave the floor to Bill. So we will have 1 hour, so we'll probably finish a little bit early. So if you have some question, we can have a brief discussion, and then after we will have lunch outside where we can continue to discuss. Okay, please, welcome.
Thanks, Ricardo. Okay, so as Ricardo said, what I wanted to do today is to talk about the connection between theoretical ideas regarding neural networks, mostly coming from the physics community, and networks of real neurons, what you actually find in the brain. And I think it's an exciting time, some of that excitement. So let me just dive in. Also, this is deliberately a bit pedagogical, so I hope that's useful for the students in the audience. We can't do this with everybody online, but this group is of a size where if something's really bothering you, please interrupt me. Nothing bad will happen, I assure you. It's not, oh, thank you. It's not that I've gotten so old that I think 1943 is recent, but I wanted in giving a review of recent progress, you should remember that neural networks has a long history. Perhaps for many of you, the subject begins. Is there a way to close this? There must be. For many of you, the subject probably begins around 1982 with Hopfield's famous paper. Let me back up a little bit. Already in 1943, which I'll remind you is after Turing, but before anybody had actually built a real computer, there was this remarkable paper by McCulloch and Pitts that proposed a kind of model dynamics for networks of neurons. And the only thing that they took from the behavior of real neurons was this all or none character that neurons generate action potentials. And so either they're on or they're off in any small little window of time. And so they described neurons with binary variables and gave a simple rule for how to update things. And what they discovered was that hidden in the dynamics of these models was essentially universal computation. So the good news was that by making a cartoon of the dynamics of the brain, you could imagine building an arbitrarily powerful computer, which sounds like a kind of beautiful theoretical result. On the other hand, you realize that if you've built a model whose dynamics can compute anything, there's nothing more for you to predict, right? Because if it can compute anything, then you can't say any more, right? And that somehow is a little bit unsatisfying. So the history of the subject then is to take these dynamics and simplify them in various ways. There are one way to think about the simplification is where the only structure in this model is the matrix of connections Jij, which reflects the way neuron J provides input to neuron I. And there are two possibilities. Either JIJ is completely asymmetric, so that the structure is feed forward, right? The neurons come in layers, one layer influences the next layer, but you never go backward. Or alternatively, things are completely symmetric. The idea that things are completely feed forward goes back to circa 1960. I always like to point out that the classic paper actually appears in reviews of modern physics, so it really was a physicist subject from the beginning. And this is where the perceptron comes from. And if you read this paper, you will find drawings like this one, in which a collection of inputs goes to a layer of neurons, which then goes to another layer of neurons. And well, there would be many more layers by the time we get to the modern era, but a lot of the basic structure of modern neural networks was already there. It's really quite amazing. As some of you know, the subject went into a dark period. Oh, sorry, there was a little bit I skipped. The mathematician Norbert Wiener was very strong on the idea that many of the things that happen in the brain in networks of neurons are collective. He was very influenced by measurements on the EEG and other macroscopic behaviors of the brain. And so he thought that there must be a statistical mechanics, that was the right way to think about it. Although he didn't get very far, but it's sort of, it's interesting. Also, it's interesting seeing that coming from a mathematician. The idea of using statistical mechanics more explicitly comes in the 70s and 80s and really gets its huge impetus from the work of Hopfield, who instead of simplifying by assuming that connections among neurons were feed forward, he assumed that they were completely symmetric. Neither of these is true, but they're useful for getting started. And if the connections are completely symmetric, then the dynamics can be thought of as a walk on an energy surface. And that, as many of you will know, allowed to understand things about associative memory and so on. Okay. So, okay, well, that worked. Maybe this also works good. Okay. So the reason that the subject is different today, one of the reasons the subject is different today than it was in 1982 is that in 1982, if you wanted to know something about the brain, what you did was you monitored the electrical activity of one neuron at a time. And of course, there are billions of neurons in the brain. There's 100,000 even in a small fly. So this is a very limited activity. The fact that we learned an enormous amount from this is already, I mean, that it's interesting why that should be true. But over the course of the last couple of decades, there's really been a revolution. One idea which started in thinking about the retina was to say, well, if I can put one electrode into the brain and put it next to one neuron and pick up the electrical signal from that neuron, maybe I can put lots of electrodes in and pick up the signals from lots of neurons all at the same time. So in general, that's a complicated thing to do. So people said, let's start with the retina, because the retina, after all, is more or less flat. So I can build an array of electrodes on a chip, fabricated with modern fabrication techniques, and then lay the retina on top of it. The signals, well, the important thing to notice if you look at a figure like this is that the green circles are the cell bodies of neurons in the retina. The black circles, it's not very visible, I'm sorry. The black circles are electrodes, and if you count in the densest region, you have as many electrodes as you have neurons, so you have a chance of picking up and sorting out all of the signals. This shows you the raw electrical signals from several of these electrodes, and there are actually two colors here. One color is the raw electrical signal. The other color is a reconstruction of those signals on the assumption that each single neuron either fires an action potential or does not. And when it generates an action potential, it always has the same shape. So what you see on the electrodes is just a weighted sum of signals from all of the neurons. There's obviously an interesting applied math problem of how you deconvolve what you see in the array of electrodes to assign it to individual neurons. By now, you can do this not just in flat systems, but in 3 dimensions with using much more sophisticated fabrication methods. And you can do this both with rigid electrodes that go into the brain and also with electrodes that are essentially made out of plastic. So, right, this is one of these miraculous things that happened in the background, is that you can make electrical measurements using things that are made out of plastic, which you couldn't have imagined 20 years ago. Well, some of you weren't here to imagine it 20 years ago. Never mind. You'll take my word for it. And so what this means is that, okay, here in a little patch of the retina, you can monitor the activity of perhaps 100 or 200 neurons simultaneously. And importantly, you can capture all of the neurons in a small patch. So what you're monitoring is all of the signals that go into the brain about some small patch of the visual world. So you get everything that the brain does, which is pretty amazing. Here, because you're sticking things into a 3-dimensional tissue, you capture only some fraction of the neurons, but the total number of neurons can reach into the thousands. A completely different approach is to genetically engineer an animal so that every neuron, or selected neurons, depending on which type you're interested in, produce a protein which is fluorescent, and its fluorescence changes in response to electrical activity. Now, the ideal thing to do would be for the protein to insert itself into the membrane and change its fluorescence directly in response to the voltage, to the electrical signal. Those proteins exist. They're not quite as good at reporting on the voltage as you would like them to be. So much more, although that's changing. So a much more common technique is to have a protein that sits inside the cell and changes its fluorescence in response to calcium concentration. Every time there's an action potential, calcium rushes into the cell and then eventually gets pumped back out. So if you monitor the calcium concentration, you have a sort of low-pass filtered version or smoothed out version of the electrical activity. So you give up some time resolution, but what you gain is that monitoring electrical activity becomes the problem of taking a movie under a microscope. And conveniently, there was also enormous progress in optical microscopy. So here is an example of about 1,500 cells in the hippocampus of a mouse. And you're monitoring this as the mouse runs around. Now, you can actually do it as the mouse runs around literally. But the other thing you can do is to combine this with this style of experiment, which is introduced by my colleague David Tank, where you have the mouse with his head held fixed so that you can put a fancy microscope up on top of it. And he's running on a styrofoam ball, which is levitated by a column of air. So as he runs, he doesn't get anywhere, but the ball rotates underneath him. You use a different kind of mouse to monitor the rotation of the ball, and then you feed that back to generate virtual reality for the mouse, so he thinks he's running through the world that you created for him. And this is now a standard technique, which allows you to combine. So the reason you need to do this is you may not be surprised to learn that there are a lot of interesting things that happen in the brain that don't happen unless the animal is actually doing something. So you want the animal to be doing something, but you also want to look in the brain with a microscope that's precise enough, that has high enough resolution that you see individual neurons. And you actually have to do better than that, because you have to be able to distinguish two neurons that are right next to each other, and they're very densely packed. So although neurons might be 10 microns across, and you say, well, seeing 10 microns under the light microscope is easy. That's true, but if you blur things with 1 micron resolution, then you just see something smooth and you can't find the cells. So you actually, in this case, so that there's the experimental physics of getting the microscopy to work. There's the molecular biology of genetically engineering the animal to produce the right protein. And then there's a kind of practical computer science of getting the virtual reality to work. I'm a theorist, so I can tell you that it's quite remarkable. I can tell you that it's remarkable that people can do all these things at the same time without, I mean, I'm not bragging, it's my friends who managed to do this. I continue to be amazed that this works. Okay, so what this means is that today it's not uncommon to visit the laboratory of one of your experimental colleagues who's recording from 1,000 neurons at once instead of one neuron at a time. And the preparations are stable. So you can certainly record for an hour or for the whole afternoon. And if you're really ambitious, you can ask yourself whether it's possible to come back to the same region of the brain tomorrow and see the same neurons and bring them into register. And with some caveats, people can sometimes do this over a period of months. So the whole experimental situation has been revolutionized. So how do you make a connection between these statistical physics ideas and the data that's coming out? So let's be precise. We're going to imagine that the neurons have some state at each, individual neurons have a state at each moment in time. There are capital N neurons. Conveniently, the states are naturally discrete because there are action potentials, so they can be binary. And one question we could ask, which is of course not the only question, but let me just to be fix ideas. You can ask, what's the probability that you'll see an arbitrary pattern of activity? So neuron number 1 is active, neuron number 2 is silent, neuron number 3 is silent, neuron number 4 is active, and so on. It's this n-bit binary word that describes the current state of the network. Can you tell me the probability distribution out of which those words are being drawn? And that probability distribution, the structure of that probability distribution, encodes all sorts of things about what the network is doing. So we'd like to understand the shape of that distribution. And it's worth remembering, one of the things you learn in statistical mechanics is that after all, equilibrium statistical mechanics begins by writing down the Boltzmann distribution. So all of the hard part eventually comes after that, right? So in thinking about networks of neurons, we're still trying to do the first part. We're trying to see, can we write down something that we believe is a good approximation to the probability distribution that real neurons obey? These questions will not be explicitly dynamical because the questions about the state of the system at a single moment in time. But of course, that's not because dynamics isn't interesting, but it's just because you have to start somewhere. So let me remind you, you can't measure this probability distribution, right? If you have 1,000 neurons, so if you have 10 neurons, then there are 2 to the 10th possible states, which is 1,000, which is not so bad. Maybe you can do an experiment that's long enough that it visits every one of the 1,000 states many times. You measure the probability. But if you have 20 neurons, it's 1,000,000. 30 neurons, it's 1,000,000,000. And at some point, the number of states that the system would have to visit exceeds the natural lifetime of a PhD student, right? And then at that point, you can't measure anymore. So there's an alternative to say that there's some features of the data that you really think are important and are characteristic of the behavior of the system. And you measure those things, they're averages over your experiment, and you say that when I write down the probability distribution, I really want to be sure I get those things right. And there's some list of them. On the other hand, you want to be sure that the model you write down, in addition to getting these things right, isn't more complicated than it needs to be in order to get them right. So let me point out that as a theoretical physicist, I'm usually in the opposite business. Usually what we do is we articulate principles that we think are relevant to nature, and we massage the data until they fit together with the principles, right? Here we're doing the opposite. We're saying there's some features of the data that we take very seriously, and then we want to take all of the structure out of our models until the only thing that's left is what's needed to explain those features of the data. And what does least structured mean? It means that if you pull the state out of the probability distribution, it will be as random as it could be, except if you pull out many states, the averages come out right. And so many of you will know that that statement in words has only one mathematization, which is that if you want the state to be as random as possible, the probability distribution should have the largest possible entropy. And so the mathematical problem you're faced with is you want to maximize the entropy of the probability distribution, but you want to be sure that the expectation values of the things that you care about equal the things that you see in experiment. So the way you do that is with Lagrange multipliers, and the answer has this form, which you realize looks like the Boltzmann distribution, where the energy is a sum of terms, one for each of the constraints that you decided to impose. And there's a constant here for each of those, and those constants have to be adjusted so that this experimental constraint is satisfied. And of course, that's in general a hard problem, which I'm not going to talk so much about. So because of that, this is a kind of formal solution, but the fact that it looks like equilibrium statistical mechanics is very useful because it gives you lots of tools for calculating things and also gives you a certain amount of intuition. So it's Boltzmann-like. As I said, the effective energy has one term for each constraint. You should notice that the statement that the probability distribution looks like the Boltzmann distribution is not the statement that the system is in thermal equilibrium. In particular, the dynamics doesn't have to correspond to a random walk on this energy surface. Right? In fact, we haven't said anything about the dynamics. So what features of the data would you like to constrain? Well, if the neurons were active more often, presumably they'd be doing something different. So one thing you might say is I should constrain how active is every neuron. So I should compute the expectation value of this variable S of I. So look, when I take a snapshot, what's the probability that neuron I is on? And I should get that number right. But then if you only do that, you haven't told me anything. So one possibility is to say, well, you can measure the correlations between pairs of neurons. So behind this is your intuition. If they're connected by synapses, if they're connected by synapses, that causes the activity in one neuron to be correlated with the activity in the other neuron. If you could get that structure right, you'd be getting something right about the global dynamics of the network as a whole. Although it's not clear that, that's an intuition, doesn't mean that it works. So you could say, let me try to get these two things right. So now let me turn the crank on what was on the last slide. What you get is a probability distribution that looks like this. And you'll notice that this looks like the Hopfield model, where the energy function of the Hopfield model has been promoted to the energy function in a Boltzmann distribution. That this model appears now not because it's some approximation to the dynamics or because I made it up, but it appears because it is the least structured model that's consistent with these aspects of the data. And furthermore, you're not free to make up what these connections or thresholds are. They have to be the ones that generate the observed properties of the particular network that you happen to be looking at. And as I say, that problem of going from your observations back to these parameters, this is the inverse of the usual thing we do in statistical mechanics. Usually in statistical mechanics, we would give you this model and ask you to compute these things. Here we have to do it the other way. So this, there's sort of a whole industry of trying to do inverse statistical mechanics. So this idea is now more than 20 years old. There was this quite nice paper in 2000 where people realized that, and actually in preparing this talk, the first version of this talk, I realized that I know many more of these people than I realized, and I had forgotten. I remembered the paper and I remembered the first author, but I'd forgotten that this list of people near the bottom were people that I know quite well. So it's interesting how you can work on something and even your own the dynamics of your own memory is interesting in this context. Let me give you the following problem. You record some free neurons, and you observe that neuron A is correlated with neuron B. So they tend to fire together. Neuron B is correlated with neuron C, and neuron C is correlated with neuron A. And I give you numbers for these correlations. And then I tell you that all three of them fire together with some frequency. The question is, should you be surprised by that, given that you measured all the pairs? So the answer is you should construct the maximum entropy distribution consistent with the measurements on this in 2000 as actually the answer to that particular problem, which then allows you to ask questions about the importance of higher order interactions in networks and so on. Okay. I think what we did was to realize that this is a path to building an approximation for the entire probability distribution, which is a little bit more ambitious than just asking about triplets of neurons, but the data were changing, and we were kind of anticipating that you would see recordings from more and more neurons, so we wanted to try and do this. So what I'm going to tell you about is basically sort of fast forward 15 years or so to detailed tests of this idea. So how seriously can we take this as a model of the real distribution? I'll say something about what I think of as doing more and doing less. So this is somehow, this approach is both too much and too little in ways that I'll make precise. And we have ideas about trying to analyze data using ideas borrowed from the renormalization group, which are somehow related to this, but help us get to much larger networks. So I'm going to speak in the editorial we, and by now we is actually quite a lot of people. This has gone on for a long time. There's several generations of students and postdocs, as well as a collection of experimental colleagues who make this all possible. Let's see, 1, 2, 3, 4, 5, 6. So yeah, so half of these people are now faculty members in their own right, and have their own students and postdocs. So this has been going on for a while, and it's been an enormous amount of fun. And I hope I can explain to you why we're excited at the moment. Let me start by remarking that when we started doing this, and I think in the evolution of the subject, there are several reactions that people have. One is this couldn't possibly work because somehow we're imposing a kind of physicist-style simplification, and we're not trying to build a model that reminds us of a real network. We're actually trying to build a model that describes the network that you actually have in front of you that you're doing experiments on. And so that's not going to work, right? Because these physicist models are too simple. The other idea is, which is related, is maybe it works, but whenever it works, that's a sign that it's not interesting. Then there are the people who think it should always work. I don't know. I don't understand either of these people, actually. The arguments that it can never work, I think, are just wrong, and the belief that it should always work seems to be misguided. As a theorist, you should never believe that your model is guaranteed to work, right? That seems like a strange thing to do. And of course, there's infinitely many different things you could have done. And in some ways, this is hard to do. So you might object that you're making your life difficult for no good reason, except for the spirit of the maximum entropy idea, which is to try to build models which are as simple as possible in a very specific sense. Okay? So that's the... All right, so let's return to the mouse hippocampus. You've already seen this. This is an example of the data that come out. This is the fluorescence signal that's assigned to a single neuron. What I want to draw your attention to is that it really is naturally discrete. It's either on or off. And you can make this a little bit more precise if you analyze the detailed forms of these things. Mostly you just threshold them, but if you are really careful, you know something about the dynamics of these indicator molecules. And so if you see it starting to turn off, but it turns off more slowly, that's probably because the neuron turned back on. But that's a detail which, when we first did this, we were very much enamored of, but I don't know how important it is. So you can say, well, look, it comes out discrete. Cells are naturally on and off. Again, I'm a theorist, so just to make you appreciate our experimentalist colleagues, I will draw your attention to the signal-to-noise ratio. Okay, this signal is raw. The little black hash is raw, so it hasn't been cleaned up. It just naturally comes out with the signal-to-noise ratio. So you really are measuring, and that comes out that way in every cell. Okay, it was really an incredibly clean experiment. So this is how you go from the image, you circle a single cell, you assign the fluorescence, you integrate the fluorescence from that cell, you discretize it, and now you have your binary variables. And you now go through the whole construction that I just described. You measure the average activity of a single neuron, you measure the correlations between pairs, you construct the maximum entropy distribution, you check that you actually have constructed it, right? That you haven't done some overfitting or whatever. And now, Does the model work? Well, in order to test the model, you have to calculate something that didn't go into constructing the model. And remember, constructing the model, we use the mean activity of each neuron, and we use the correlations between pairs. So we need to do something beyond pairs. So an example is you could ask, what's the probability that K out of the n neurons are on at the same time? And in this case, it's a population of 100 neurons from a small patch inside that large window of 1500. And you see that we get the answer right quite accurately. And let me point out that after this, right, a nice feature of the maximum entropy construction is that once you've matched the things and the data that you want to match, there are no adjustable parameters left. So everything that you calculate is a parameter-free prediction. In fact, you can think of the maximum entropy formalism as a kind of engine that takes some features of the data as inputs and makes predictions for other features of the data. Just sort of what we're always doing, but in this case, the steps are very clear. And so that also, I think, is some conceptual advantage. Remember that this is a model that looks like statistical mechanics. So there's this object in the exponential of the probability distribution that looks like an energy. So you could ask, well, what is the distribution of that energy itself? Which is sort of the probability that you find the network in a state with a certain probability, right, or a log probability. And indeed, you get that right as well. Let me point out that you're getting it right out to energies which are of order 30. So e to the minus 30 is what, 10 to the minus 15 or so, 15 or 20. These are very low probability states. Of course, we don't know whether we got the right states assigned with this probability. We just know that we got the right number of them, right? That's all you can test. But you get the details right, including these funny little wiggles that you might have thought were noise, but they're not. That is to say, the error bars on them are smaller than the wiggles. Yes? Absolutely, yeah. Yeah. So, right, so one question you might ask is, if you measure, so we're saying we match the mean activity and correlations. But you say, well, is that over the whole experiment? Is it over part of the experiment? So the first question you might ask yourself is, are those things consistent over subsets of the experiment? And the answer is yes. The experiment's about an hour long, and the animal's healthy, so it's not such a problem. And he's adjusted to the environment and so on, so the animal's not learning over the course of the experiment. And then when you predict these things, you always predict and test them on parts of the data that you did not use in construction of the model. Although because you're computing something which is higher order, it's not obvious that there's an overfitting problem, but just to be safe. In earlier work, we actually did this, being very careful about sizes of the fractions and so on. Okay. Maybe we shouldn't have been so fancy. We matched the correlations between pairs of neurons. So you might ask yourself, well, why don't you just check the triplets? And in fact, with 100 neurons, there are more than 100,000 distinct triplets. If I showed you a scatter plot of the results, it would be hard to read because it would be a dense blob with 100,000 dots. So what you could do, for instance, is to take the axis of the triplet correlations, make little cuts along the axis, and then in each bin, you could get the average of your prediction and its standard deviations, and you see that they fit very well. If you're a little bit more ambitious, you could ask in each bin, what is, like, so somehow I put observed here, but where is the error bar in my observation? It's hard to figure out where to put it on this graph. I know what the range of predictions is because I have a little bin, but what I could do is say inside that bin, there's some root mean square error of my predictions, which is what's shown in red here. But then I can also take all the data points in that bin and ask, what's the root mean square error in my measurement, which you get in the standard statistical way. And what you see is that those are equal to each other across this whole dynamic range. So that means that we've predicted 100,000 things within error bars, across their whole dynamic range, which we found quite surprising. The other thing you can do is to say, well, look, this is a model of collective behavior. So what that means is that if I tell you the state of all of the neurons except for one, then I can compute the probability that that neuron will be active. And so what I can do is to go through the entire experiment for every neuron and for every moment in time and compute what in the language of the underlying Ising model is the effective magnetic field acting on that one neuron. You collect all the moments when the effective field contributed from the other neurons has a certain value. You then ask, what is the probability that you find the neuron to be on at that moment? And you get the red data. And of course, if the model were correct, you'd get the, well, shifted hyperbolic tangent, which is the thing that's the dashed line you say they agree. This is somehow averaged over neurons and over time. More interesting might be to unfold this in time and to see what happens as the animal is running in this virtual world. And what you see is that, well, so here's the activity of the actual neurons. And you see that what happens when the animal runs through the world is that the neurons fire in sequence. That's because they've been arranged. This is the hippocampus. And the hippocampus is this magical part of your brain that has cells that are responsive to where you are in space. And so if you're running along a track, then I can put the neurons in order. This is the one that's responsive at the beginning of the track, a little ways further down, a little ways further down. And if you order them that way, they will fire in sequence in time. And that's what you see. And an observation that goes back almost to the discovery of these so-called place cells is that sometimes neurons miss their place field. Right? So you notice that when you're here, they all fire in sequence. Here, these guys dropped out. And so usually the view is, well, you know, the brain is noisy. But remember, we can compute the probability that each of these neurons should be active, given all the other neurons in the network are doing. And that's what's shown here. And you'll notice that given all the other neurons in the network are doing, these neurons are predicted not to be active, and they are not. So what we thought was noise is really collective dynamics. And well, we don't know what it means to the animal, but we know that it's not just a randomness in your response to the environment. It's a reflection of the internal collective behavior of the network. So that's great. We can give these quite precise predictions for populations of 100 neurons. We basically get everything right within error bars. But if you're recording from 1,500 neurons, you could have chosen those 100 neurons in many different ways. So what we did, everything I was telling you about is you choose them from a small region. So they're near each other. At the opposite extreme, you could choose them at random from the entire region. So the little red dots here. And if you really want to be careful, you could make sure that if you measure the average activity of each one of these 100 cells, the distribution of that activity looks the same as over here. And in fact, if I showed you the distribution of pairwise correlations among these neurons, it looks the same as the distribution of pairwise correlations over here. So if I didn't tell you where the neurons were coming from, and I just showed you the data, I would say, I don't know, it looks about the same. I suppose if you construct the model, you'll get the same answer. And the answer is no, you don't. So here are 3 examples from the tight to the completely scattered. This is the thing that I showed you before. And let me just jump to the end. If you try this in this from neurons that are randomly chosen out of this large area, it's kind of a disaster. Now, I should point out that once upon a time, if I told you that the error bars of your predictions were within a factor of 2 of the error bars of your measurements, you might have said, well, you know, this is biology, that's pretty good. But in fact, you can do almost perfectly. And similarly here, you might have said, well, I don't know, I capture the general trend. But in fact, in a different population of neurons, you get everything exactly right. And finally here, I think everybody would have said that these bumps and wiggles are just noise and trying to get them right is hopeless. But in fact, they're not noise because we can assign error bars and their variations are much bigger than the error bars. And if you choose the population correctly, then you do get it right. So what is this telling you? It's telling you that at least in this system, when you're describing a compact region of the network, which presumably has most of the interconnections, then this description in terms of simplified models really works. But if you start grabbing neurons at random from a much larger region, they're interacting with all sorts of other neurons that you're not seeing, probably. And so as you average over those, you effectively generate higher order correlations, higher order interactions among the neurons you're looking at, which you can't find out about by just measuring the pairs inside the population. And so this is sort of from between the people who thought it should always work and the people who think it should never work. This is sort of the right answer, right? That it can work with really remarkable precision, but only if certain conditions are met in the network. So success tells you something about the underlying structure in the network. I would also say, so this point that, if you hadn't seen what real success looked like, you might think that some of the things that were failures weren't so bad. And what I would say is that, if we have this conversation 10 years from now, it's very likely that the kinds of theories that we're talking about look nothing like this. I don't think our achievement was to make these theories work in this context. I think our achievement was to show that the quality of the experiments is now high enough that you should take theories seriously as making quantitative predictions, and you should expect that you can get things right within the error bars rather than just capturing trends. Now, if you come from physics, of course, this sounds perfectly sensible, but you realize that in the context of biological experiments, there was a tendency to raise your hands and say, well, biology is complicated. You can't really hope to get everything right. Yes? So is it reasonable Yes, which, is not a crazy distance given the structure of the neurons and things like that. So it is particularly interesting to have this conversation in the hippocampus where for some reason there's a long-standing debate about the nature of interactions inside the region. There are two parts to the hippocampus. Okay, it's complicated. So an important tension here, which is maybe if I can generalize away from the question for a moment, every region of the brain has its professional explorers. And so in some ways, an enormous amount is known about this region and about this region. And of course, the people who spend their lives studying one of these regions, the things they're most interested in are what makes it different from the other regions. And so you somehow have to be able to touch that with these theoretical structures, but maybe it's not interesting to talk so much about that in a colloquium like this. So that's one of the things that we have to deal with. So I haven't talked so much about place cells. Where are they in this discussion? That's a whole nother topic. Yes.
Sorry, please.
You said that the measurements of the pairwise correlations were fairly robust and stable across different.
Yes.
But are they also robust in terms of distance between the pairs? Like when you look at the pairwise correlations in between neurons that are far away or pairwise correlations of neurons that are
for example,
are they similar?
You can find, so if you look at the distribution of pairwise correlations among these neurons and the distribution of pairwise correlations among the red neurons that I've marked here, those distributions look the same. There is some tendency for neurons that are closer together to be more correlated, but it's not a very strong tendency. It's actually not even a terribly strong tendency in the retina, by the way, where you might think it's completely obvious that neurons that are next to each other, they're looking at the same thing, so they should be more correlated with each other. And yeah, vaguely correlations die off with distance, but you can see neurons that are very close to each other that are not correlated, neurons that are very far apart that are correlated.
So the discrepancy in the sparse case, it's more about the sparsity of the sample.
We think it's about the sparsity, because in some sense, right, if the interactions are relatively local, if you sample sparsely from a large area, there's lots of things interacting with the neurons you're trying to describe that you're not seeing. And so that you can't get all those things just by measuring correlations among the pairs.
Like, could it be possible to replicate this discrepancy between a dense sample and a sparse sample theoretically? Like looking at what happens if you have a really large points with those pairwise correlations.
So I think
then you look at a subsample and you reach to the
I think you heard the answer, right? If I take a system in which I have relatively local interactions, and I densely sample a local between pairs, and I densely sample a local region, the pairwise correlations that you measure in that local region provide you with enough information. They're not exactly the same thing as the interactions, but they provide you with about the same amount of information as if somebody had told you the real interactions. But if I now start grabbing things that are very far apart, well, this guy mostly interacts with neurons here that you're not actually recording from or not using in your analysis. And so most of the interactions that are relevant are not being monitored. And so these two cells, they interact with each other, but there's many numbers that go into contributing those, to determining those interactions. And if you have three cells like this, then you can have an effective triplet interaction that comes from all averaging over all the other neurons, and you have no access to that by looking at the pairs. Good. Please.
If you still pick a group that are of neurons that are still packed together, but they have a different shape such that the boundary between the non-observed neurons and the observed neurons is longer.
Oh,
Do you think you get worse approximation?
so that's an interesting question. Right, in some way, you should be able to learn something about the shape of the interactions, right? That, you know, for instance, if for some reason the network had an axis, then presumably, you know, capturing neurons this way would be different than capturing neurons that way. So probably yes, but we haven't done it. And actually, this is probably not a very good place to look for that, but so be it. Good. Okay. So in the interest of time, I'm going to be a little less ambitious and try and explain clearly what comes next and skip the part about renormalization group methods because it's kind of a whole other structure. So let's stick with this idea about using maximum entropy models, but let's be a little more careful and look at more systems. I've shown you an example where things work really, really well. Sometimes they don't. And so an example from some years ago in the retina was a situation in which things didn't work so well. And actually the sign that they weren't working very well was interesting. I showed you how in the hippocampus we predict the probability that K out of N neurons are active at the same time. If you tried that in the retina, already when you had 10 neurons, it worked quite well. But by the time you got to 20 neurons, you were already off by a few percent in predicting the probability that the entire network was silent. So you might think to yourself once again, right? The reflex is, well, you know, it's biology, things are complicated. Why are you upset about a few percent discrepancy? Well, the reason you're upset about a few percent discrepancy is that the network is silent a lot of the time. Action potentials are rare. So in fact, in a network of 20 neurons in the retina, the probability that the entire network is silent is, first of all, not very small. It is actually the most likely thing is that the whole network is silent. And therefore, if you record for an hour, you have a very accurate measurement of that probability. And so if you get it wrong by a couple of percent, that's actually a big error, which I can tell you came as a surprise to many of our biological colleagues when we told them that a few percent off is a big error. So we tried improving on these models, and indeed you can do better, but I would say that this whole story is not terribly satisfying. We're clearly missing something, and one path is to add additional constraints in the spirit of a maximum entropy structure, but it suggests that somehow we're not doing the right thing. So I wanted to leave that for you as, you know, it doesn't always work as well. And in fact, I would say, now that we've seen how well it can work, we're even less satisfied with how it worked before. At the same time, you realize that trying to match all of the pairwise correlations, if I have 100 neurons, then there are 5,000 pairwise correlations. You record for an hour. You have many tens of thousands of samples. Everything's okay. But as I increase the number of neurons that I can record from, but I don't increase the duration of my recordings in proportion, I very quickly get into a state where it's not that I can't match all of the pairwise correlations, it's that I don't actually know all of them because I haven't collected enough data. So I remind you that if you measure a correlation matrix, right? If I have n variables and I measure a correlation matrix or a covariance matrix, if I have t samples, the rank of the matrix can, measured from the data, can never be larger than t. And so if t is less than n, you're done. You haven't actually measured the covariance matrix. So the idea of constructing a model that matches it is sort of wrong from the beginning. It's not a computational problem. It's that you don't actually know all these things. So in some way, if you want to take this approach to larger and larger systems, you're going to have to add not so many constraints. You're somehow going to have to decide that you don't match everything. You match some number of things that you believe you know for sure and stop. So there's a very nice paper recently about how to do this. And what they said was, well, suppose that the things I match, I imagine that there's a neuron that's looking at the network. And what it does is it takes a weighted sum of the inputs, compares it to a threshold, and computes some nonlinear function, just like the standard model for what neural networks do. And it is these things that I try to match. And you can decide how many of them you want to match. So that's good, right? Because then you have control over how many features of the data you take seriously. What was interesting was that when they started, they realized that they could pick these coefficients at random and do not too badly. So you can compare, for instance, this is in a population of neurons in the visual cortex. If you build the kind of pairwise model that we were talking about, this is actually in a regime where you can do it, then your performance in terms of the likelihood of the data in the model would be sitting here. And if you take these random projections, you can, by the time you have a few thousand of them, you're doing even better than the pairwise model. The pairwise model, of course, has 20,000 things that you're matching. So this is interesting. You can actually do better by not choosing at random. So this paper is interesting because it's a little schizophrenic, right? So on the one hand, they say it's fantastic that you can do so well with random matching. But then actually, if you try to optimize these things, you can do even better. But this is a hint that it's not that the spirit of maximum entropy was wrong. It was that the particular constraints that we decided to choose, we made them up, right? There's a historical reason why we think pairwise correlations are important. But who knows? Maybe we should pick something else. So this was a very different idea about what kinds of things you could pick. And you see that it works, extremely well or surprisingly well. So this opens a completely different direction. Let me take another direction, which my colleagues and I have been working on. And this direction says, well, okay, let's stick with pairwise correlations. But of course, since I don't know them all, I can't match them all. So let me pick a subset of them. Which subset should you pick? Well, the answer is you should pick the strongest correlations, right? Because that tells you the most about what's holding the network together. Well, how do you find, you don't actually want to pick the strongest correlations. What you want to do is to pick the correlations that when you impose those as constraints, they the entropy of the model by the largest possible amount. Because that's what corresponds to capturing more and more structure. The problem is that you can't find those things. You'd have to test them all. And it would be combinatorially hard. Unless, of course, the correlations that you pick fit on a tree. Because if you fit on a tree, then the easy model is exactly solvable very quickly. And you can test. And furthermore, you can actually analytically compute the effect of adding one more constraint. So as you build up the tree. And there are theorems, this is like the minimal spanning tree problems in computer science. So there are problems that show you that you will find the optimal tree by a greedy algorithm. So you put the two neurons that share the most mutual information together, you then find a third neuron that when you attach it, it's still a tree, which just means it's not, well, actually with two, you have to make sure it only connects to one and not to the other, right? And then you pick the one that has the biggest mutual information with the thing that you're going to attach to, and so on. You walk through, and in seconds, you solve the problem for networks of thousands of neurons. The only problem is that you built a tree, and who told you that the interactions were really tree-like? And the answer, of course, is they're surely not. The question is this the beginning of a good approximation scheme? So how do you tell whether it's the beginning of a good approximation scheme? You try computing something collective. And so an example is to choose this probability that k neurons are active at the same time. And here you've built a model for a network of more than 1,000 neurons. If you pretended that the neurons were independent, of course, you'd see an approximately Gaussian distribution. And instead, you see this thing that has long tails, and you almost get the tails right just by building this tree-like model. So one clearly captures something about the collective behavior of the system, even though you're only searching through this very limited set of possibilities. So it's about time to stop. So I'm going to stop here. And let me try to summarize. There's a strategy for building models, which says take some features of the data seriously and strip out all of the unnecessary structure. It has a certain coherence and logic, and it's appealing. And it produces models that we understand something about because they look like statistical mechanics models. It's not a coincidence, right? The Boltzmann distribution is the distribution that has maximum entropy consistent with the average energy. Within that class, we've seen how in real systems, we can get descriptions which are accurate, essentially reproducing every feature, all the statistics, higher order statistics of the network that we've tested within experimental error for real networks, despite the fact that the structure is very simple. We also see that that has its limits. Sometimes it just doesn't work well enough and we're clearly missing something. In other cases, we want to reach to describe much larger networks. And so we're going to have to ask more carefully what features of the data do we think are the essential ones that capture the core characteristics of the network dynamics. And so there have been at least two different ideas. One is to imagine neurons that are downstream reading this out and say that what you care about is the activity of those hypothetical neurons, which I think has a certain appeal to it. And the other idea is, well, just do what you were doing before, just don't take everything, try to put them in some way and be selective. And I'm sure there are more ideas, but that's a sense for where we are. And it's driven by this interaction between theoretical ideas that reach back before most of you were born and experimental techniques that are rapidly changing and making all of this possible. So thank you.
Well, thank you. Thank you very much. We have, I hope the students have appreciated the talk. I mean, maybe some of the things were not clear, but at least you have, an understanding of the contact with the research. So we actually did maximum entropy in class. So,
So if you didn't understand, it was his fault.
So it's time for one or two questions, and then we have lunch outside. So, are there any questions?
A feature of your modeling is that the interactions between neurons are symmetric.
Yes.
And still it works very well. So is there a way
Going on, yeah.
I mean, is there a way to include some asymmetry?
Yes. So the interactions are symmetric because we're describing the distribution of states that you see at one moment in time. And that's symmetric, right? As soon as I ask you about sequences of states, then you have the possibility of asymmetry. And so I myself have not done much with this, but some of my colleagues have. And then of course you get, if you ask for if you ask to match, for instance, let's take two neurons. I ask you to match the mean activity of each neuron and the correlation at one moment in time. Then of course you get a symmetric interaction. Suppose I ask you to describe the probability distribution of the time series, and you want to match the correlations between pairs, but the correlation functions as a function of the time difference. Of course, that's asymmetric, generally. I mean, you might by chance find two neurons that were symmetric, but generally asymmetric. And then the interactions will also be asymmetric, and will give you some sense of which neuron is driving the other, if in fact that's a good description. So it's really because we asked a question about a snapshot in time that we're left with symmetric things.
So I had a related, but maybe not the same question. So these movies, when you have this experimentally, you have these mice that are watching movies.
Well, they're running around, yeah.
So maybe the stimuli they get vary in time.
Yes.
And it's like, for example, I don't know, the external feed we have to vary in time or something like that. And how do you account for that? Or
So what we did here, we essentially are integrating over those time varying external fields and treating them as effective interactions. I think it's one way to think about it. There's a long tradition in neuroscience of trying to separate the correlations between neurons into components that are internally generated and components that are inherited from the outside world. So the generic answer is it's half and half. And we could discuss why that turns out to be true, but it's actually not that surprising if you think about it. I think. So that translates into I convinced myself it made sense once, right? There have been ideas to take the time series of responses in cases where you can. So, for example, when you're showing a movie, you can show the same movie again. And you can thus line up things in time and separate variation across time that's locked to the movie versus variation that's not locked to the movie. And so you can think about matching the mean activity as a function of time and the pairwise correlation. You could also match the pairwise correlation functions if you wanted to, but maybe that's too much. So if you do that, you literally get an Ising model with a time-dependent external field. But the interactions are still non-zero. One of the students in the group, Caroline Holmes, has been working on this, not with me, but with our colleague Stephanie Palmer, and experiments that were done by Olivier Mar's group in Paris in the retina. And one of the things they're finding is that they can actually do that decomposition, and the interaction matrix that you find is the same under very different conditions. So nobody knows how to turn the movie into time-dependent fields. That's terribly complicated. And there's no unique answer because the retina is adapting to different environments. But it seems like the interactions have some transferability from one experiment to another. So that path that you were thinking about seems like it has some real distance to go.
So maybe this question is a bit naive, but what is the use of the model afterwards? Like, is it just used for testing the hypothesis that the observable you're forcing are actually the relevant ones? What's the use of it?
Yeah, very good. And quite fair, right? Why should you care about this? So there are several answers. One is, what you've done is to create a statistical mechanics for a network of real neurons. I think I have the analog of the Boltzmann distribution in my hand. I could now ask you, where is the real network in the phase diagram of possible networks? Which I couldn't do before because I didn't have the analog of the distribution in front of me. The answer is that in most of these cases, it seems to be near some sort of critical surface. which we think is interesting. And again, some people think it's not interesting, but there's an argument to be had there. So that I think the question, where do you find, can you tell me what you mean by the phase diagram of possible networks and where is the real network? That's a good question that we can now address. You have to believe that you have a good approximation of the distribution, right? Otherwise, you get it wrong. That's one thing you can do. Another thing you can do is you can try to make contact between this structure and other things that we know about the system. So for example, in the hippocampus, there are these place cells, and we have, remember we said that you could predict when one cell would be active given the state of the rest of the network. So you could say, oh, you're just cheating. What's happening is the state of the rest of the network tells you where the animal is in space, and then where it is in space tells you whether this cell should be active. But it turns out that only 1/3 of the neurons are actually place cells in the experimental environment. That's actually been known for a long time. They're presumably, the other cells are place cells in some other environment. Otherwise, you'd run out of cells. And so the fact that this works equally well, you can sort of decompose this effective field into a contribution from other place cells and a contribution from non-place cells. And you see that those have the same order of magnitude. So what that tells you is that the network is sharing information, maybe half of which is about place and half of which is about something else. And so that leads you into asking, well, what is the something else? And okay, so that's another example. I mean, I could go on, but maybe those are two things that are very much, having constructed a statistical mechanics, there are certain natural questions from the physics point of view. And then having built these kinds of models, you can say, well, how do they connect to other things that we know about this particular network?
Can I ask you, okay. Since I asked a question, so what about other technologies that could scale up the number of neurons, like say calcium measurements or this kind of thing?
So the experiments that we showed here, which are on order 1,000 neurons, let's say, are calcium imaging experiments. You know, it's funny. The pace of theory and experiment are different. And the things that make things slow and fast are different. So I'm afraid we theorists are behind, in the sense that, you know, we worked very hard to get from 10 neurons to 40 neurons to 100 neurons to 1,000 neurons. And now I have a colleague who actually comes from the quantum optics world, but he has this beautiful thing of using the temporal structure. So when you want to do 2-photon microscopy, so you excite the fluorescent molecules with 2 photons, and you measure the 1 photon that comes out. Why do you use 2 photons? Because the trade-off between scattering and suppression of scattering and everything, right? In order to see fluorescence, you have to make sure you're not seeing scattered versions of the original light. But of course, with 2-photon microscopy, then they're already a factor of 2 apart in wavelength, so that makes it easier. But 2-photon interactions don't happen unless the intensity is very high. That's good because the intensity is high in a smaller region when you make a focal spot. But the only way you can do this is with pulsed lasers, right? That's the only way you can get to sufficiently high intensities. So if you fiddle with this pulse structure in the laser, you can actually get it so that instead of illuminating a spot, you illuminate a sequence of spots in the vertical direction. And then you scan. But the sequence of spots, right, if you really exploit the fast pulse structure of the laser, you essentially get the sequence of spots. You're discriminating sub-microsecond, in fact, close to nanoseconds, in order to tell them apart. But of course, you spend the timescales of interest are milliseconds and longer. So you're effectively capturing the 3rd dimension instantaneously. What that means is that they can get to 1 million neurons. They've tried some of the RG-related things that I didn't have a chance to talk about, but we're behind. But just to give you a sense of where the field is going experimentally, it's quite amazing.
Since you mentioned the renormalization of the flow technique, do you expect some enhancement of symmetries for some fixed point of flow?
Let me see. What's the best way to say this without starting to give the whole part of the talk I left out? When you use the renormalization group, to analyze models in statistical physics, right? So Michael Fisher is reported to have said, Ken Wilson's greatest discovery was irrelevant operators. So one of the important consequences of the animation group, right, is that in a sense that we all understand, even if it's a little sloppy, models become simpler as you coarse grain. So the idea is that by trying to systematically coarse grain the activity in a population of neurons, we somehow arrive at something which is simpler. Of course, we don't have ridiculous with data. So it's not clear whether it's simple, it will be simpler in the sense that it is more symmetric or just simpler, right? Smaller number of operators. So we don't know. What's surprising is that when we actually do this, we find all sorts of interesting scaling behaviors and so on. So that's kind of what's distracted us from the problem of constructing the models.
Okay, so one last question.
I wanted to ask the following thing. So you showed us that at least on a small scale, given those two features and using this maximum entropy principle, we were able also to explain quite well other features that we were able to measure that did not use in the training. My question would be, are there any features that did not match what was given by the model, and if yes, would it make sense to use those features in the training of the model to add the structure that we're missing?
So in the hippocampus, everything we try works. Maybe we're not smart enough to think of the right thing yet. Okay, you never know. In the retina, we found already that it doesn't work. And as I said, one particular thing that didn't work was predicting the probability that K neurons out of N are active. So that, if you think about it as a kind of global quantity, right? So usually we think, you shouldn't write down interactions that have that sort of global structure. They look funny. Except, of course, if I have a population of neurons and there's one neuron that I'm not recording from that has a synaptic connection to all of them. Well, from the point of view of this population, that's a global interaction. So maybe it's okay. So we tried it. And indeed, things got much better. So this is an interesting example, which perhaps I should have explained a bit more carefully. We put a constraint on the probability that K out of N neurons are active. So now we can't use that as a test anymore. But we still can use the triplets. But at first you think about it, you know, how do these three neurons know that we put a constraint on the global activity? And how does that distinguish these three neurons from these three neurons? Well, the answer, of course, is they still have their pairwise interactions. So they have an identity by virtue of their pairwise connections. And giving them that extra bit of global information is enough to substantially improve your prediction of the triplet correlations. So that's exactly the path you're describing. And the problem is that it's a little bit like when you do perturbation theory, you know? If you don't basically get the right answer at first order, you can try second order. If you still don't have the right answer, I don't think computing third order is a very good, I mean, you can if you want to, but somehow there aren't very many problems that were solved that way, right? So somehow if you start in a place that seems like a sensible approximation and it doesn't work, you could try improving it a little bit, but you shouldn't, you know, add too many things. You should rethink where you started. And for instance, these guys were rethinking where we started, which I think is a good, I mean, is it the right thing? I don't know, but it's the right idea. Pick something different. Stay simple, but different.
Okay, I think we can thank Bill once more. So, so there is lunch outside. COVID is over, apparently, at least, so we can finally have some social event. And I want also to mention that Bill organizes a very famous school in Princeton every year about biophysics and computational problems and for young students. So maybe in the future you will end up visiting Princeton in this school. Okay, thank you very much. That's good. Prior to the US, already at the level we yes, yeah, so yeah, we have this strange belief in breadth. High schools aren't good enough. Yeah. Right? I mean, you believe in breadth too, but you get it by making it in high school or something, right? So you know what I mean? Hey.
Do you mind if I ask you a question?
Not at all.
Okay, well, so I'm an undergrad.
Mm-hmm.
And a lot of the things that, went to the presentation, I put a lot of effort to try to understand it, but I just wanted to make sure that I did.
Yeah, yeah.
So did I understand correctly that essentially you had a mouse? You put the mouse on this machine. Yes. On top of which you have an instrument. Well, I'm trying to figure out where people can pause. Yes, start. You've got a voice of one. And you have this microscope on top of it. See. Yes. And for those of like signal, it's like creates with information, right? Creates a distribution of the files and the neurons. And so in the second or something that is. Well, no, I think you really were aiming for some sort of this. I mean, in some sense. Time, some attention. Yes, well, okay, what was the thing we did? The thing that was important was to take seriously the idea that you like that real distinction. But then before that, people would say, well, all very complicated, maybe this way or that way, good. What if can you do simple guys and work out the